Those of you working in other fields of physics, it's perhaps a bit surprising that some statistical physicists study neuroscience. This has been the case actually since the advent of cybernetics in the late 50s, early 60s, with early figures like David Wilshaw, Jack Cowan, or Leon Cooper. But it is only in the 80s that a model proposed by John Hopfield as suggesting that metastable low energy state in a disordered Ising model could explain memory that has attracted widespread attention. This was especially the case at ENS, where important contributions were made by Gérard Toulouse, Bernard Derrida, Jean-Pierre Nadal, and Marc Mézard, our present director. So, this is John Hopfield and his famous article. But I actually read Hopfield a paper in '83 when I was doing this internship in Philippe Hacher and Coco Gershenfeld Lab. I found it fascinating at the time, but the direct contact with experiments seemed remote. So I actually came to neuroscience much later by a different route that I will explain you. So a key event for me was a 5-minute conversation with Reinhard Ecole at a conference in the Canary Islands in '83. That was a conference organized by Agnesa Babloyans, who was from Université Libre of Brussels. So this was the group around Ilya Prigogine, who were very early in trying to explain physics. So I gave a talk there, and Reinhard Ecole came to me and said, okay, your talk was interesting for me because I record things that resemble what you have said. And what was it? So he was recording in the visual cortex of monkeys, and he was seeing that when he was recording a lot of neurons, like look-at fit potential is the activities of many neurons that are together, he was seeing an oscillation here in 60, 80 hertz, 90 hertz. But when he was recording, so in principle, this is coming from the activity of single neuron. And when you record single neuron, actually you don't see any peak in their frequency. They are not rhythmic at all. So that was his observation. He said, okay, it would be nice to explain it. He was a physicist, actually. And this is something that he was recording in the cortex of monkey, but is actually quite widespread. Here it's in a rat hippocampus, another region of the brain. And here you see like 200 hertz oscillation. And here what you see, maybe I should have said that, these are different traces of single neuron. Here also, these are called raster plot. And you see that this neuron, for instance, emit a spike here, 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 but not in a rhythmic fashion. And so the question is, that was asked by Reinhard, is can we get this kind of thing with neurons that are doing things like that? Which at the time I had absolutely no idea, actually. So this is an interesting question because these neural rhythms are very widespread. They have been recorded since the beginning of the electroencephalogram. So this was discovered by cat on dogs, but then Hans Berger, a German physiologist, recorded them in human. This is the first published EEG in human on the son of Hans Berger. And what Hans Berger observed is that here you have an oscillation at 10 hertz. So this is non-invasive. It's just on the skull. And so when his son was at rest, okay, it was oscillating at 10 hertz, and then he asked him to do some math problem, and it was going to beta rhythm. Okay, so after this was discovered by Berger, Edgar Adrian started to put electrode in the brain of different animals and actually realized that, okay, there are a lot of different rhythms and they depend on neural structure and activity. So this is a problem that is of some interest. Okay, so before trying to understand that, I probably need to remind you a little bit about neurons, and I had myself actually to learn a bit about neurons. So neurons are complicated things. Here you have very nice drawing by Ramon y Caral. Ramon y Caral is probably the most famous anatomist of the last 2 centuries. So what Ramon y Caral did is actually that he depicted this neuron with a great precision with a technique that was invented by Camillo Golgi that stained a few neurons. And so here what you see is a drawing by Ramon y Caral, and what is really remarkable is that he has put these arrows. So here is something that starts from the eye, so it's actually how he put the arrows, because he knows that light is coming here, so information should travel this way. So from this, he deduced that, okay, the information comes on the dendrite and goes to the axon. And so he could put, having identified this feature of autonomy, how the information travels in other parts of the brain, like here, a region that's called hippocampus, that it implies in memory or navigation. And also, Ramon y Caral discovered that neurons are really discrete cells and that are connected. He postulated that they are connected by synapse, junction in Greek. Okay, so these are the basic elements. But when you see these very elaborate things, it seems very complicated in some sense to do anything, because it's very, very complicated. You have a lot of neurons. So, and moreover, when you see that, you don't know how things work. So I'll try to explain you how things work. So the first thing is that the neuron is a cell, and so is insulated from the exterior by a membrane. And electrically, this membrane is like a capacity. One can compute very easily the capacity is like one know the width of the bilayer, and saying that, okay, this lipid bilayer is like oil. One get this almost universal number that is 1 microfarad by centimeter square. So this is the capacitance per unitary of a biological membrane. So that's one element. The other element is something that was discovered by Nernst, is that if you put 2 solutions with different concentration, and if you put a semi-permeable membrane, you have immediately a difference of potential between the 2 sides. And this is, okay, this is Nernst's law. So the difference of potential is just the log of the ratio of concentration. And so it's mechanically is rather easy to understand. If you have a lot, say, of potassium on this side, and not too much potassium on this side, and things diffuse, the potassium will tend to diffuse on this side, but then there will be, of course, the 2 solutions are neutral, so there will be incompensated charge on both sides. There will be an electric field, and things will stabilize. So this is an equilibrium phenomena with a semi-permeable membrane. One gets for free a difference of potential. One other way easier to see that, maybe for statistical physicists, is to exponentiate this relation. Is just say that the probability to have an ion on either side is just proportional to the Boltzmann factor. So here you have the ratio of concentration are proportional to the exponential of the difference of energy. Okay, so it's a simple thing. And so this, once one has realized these 2 things, then one is led to a model of a neuron that is quite simple. It's just a very simple RC circuit. Where is it? The intracellular side, the extracellular side, and this is a piece, so there is the capacitance of the membrane. There is a diffusion of ion inside the membrane, and if it's semi-permeable, there is a difference of potential. So this is the simplest description of the neuronal membrane, and this was the state of electrophysiology at the beginning of the 20th century, with a major contribution by Neurons, Bernstein, who synthesized this thing, and Lapik, who was a French, very famous French neurologist, who actually discovered this capacity. And if you actually read the paper by Lapik, you see that, okay, he was a biologist. He had some difficulty to solve this RC circuit, and he asked a student of ENS that he thanked in the paper for solving this differential equation with the single exponential release. Okay, so the neuron membrane is a low-pass filter. And then, so this is a linear thing. So the thing that was added by Bernstein is that if the depolarization is strong enough, then presumably something happens, and there is an action potential, a large excursion in potential that goes from negative at rest to positive and then comes back. So this was not really understood. It was understood later in the 50s. And then, so this is this model. If the membrane is depolarized, something happens, this action potential, and the membrane repolarizes. So this is called the leaky integrate and phi neuron. And although the action potential is not described, it's a good model to start with. So here I show you the people who did all this. In the context of this seminar, maybe we can note that Walter Nance was really a very vocal advocate against the Nazis in Germany, and actually was dismissed from his, in 1933, from his position as the Kaiser Wilhelm Institute. And you see Willa Peak here. Okay, so now we have to, so we want to describe a network, so I'll go fast about synapses. So synapses are very interesting and complex structure. And you see that each neuron is covered by synapse. Here, these are this red thing on this neuron. So each neuron has about 1,000 or 10,000 synapses. And a synapse, so what does it do? So when there is an action potential in this neuron before the synapse, the presynaptic neuron, then there is some chemical transmission at the synapse, and the postsynaptic neuron is slightly, the potential in the postsynaptic neuron is slightly modified. It's either depolarized or hyperpolarized, depending whether the synapse is excitatory or inhibitory. And you see that the tip... Ah, wait, okay, pardon. Okay, so, and so you see the typical strength of a synapse is like 0.2 millivolt, and you need to depolarize a neuron by, say, 10 millivolt to get it emit an action potential. So you need the combined action of many presynaptic neurons to get a neuron phi. Okay, so now we have the basic thing to describe a network of neurons, and that's what we do. So one can try to simulate neurons. So this, actually, people tried to simulate neurons quite early. I mean, in the 50s, people simulated neurons, and this went for quite some time. But most people simulated a few neurons coupled together, like 2, 3, to get some rhythm, etc. It's only in the 90s when people started, as Edward said, to study this model that was proposed by John Hopfield with a binary neuron, so 1 or 0. And so they make a lot of progress on studying this model, but after a while, they realized that this model had shortcomings, and they thought maybe it's not a bad idea to start and look at neurons that are a bit more realistic. And in particular, this was happening at the mid-90s, and this was in particular started by Daniel Amit and Nicolas Brunel, who was there at the time in Rome. And their idea was, okay, we'll try to understand why how can we make a neural network that fires at a low rate, like when you observe a pyramidal neuron, it would be maybe 5 hertz, something like that. So how can we make a neuron like that? And can we make memories in this network that are very, I mean, that would be the analog of what is postulated in Hopfield network. So when they were trying to do that, there was a little bit annoying feature for them, is that for some parameter, they were seeing some oscillation that were annoying. So when Nicolas came back here, he gave a seminar, I noticed that, and it reminded me of this thing that Reinhard Ecole had told me, and that I didn't know how to explain, so I thought, okay, this is a bit similar, so let's try to explain it. And so with Nicolas, we simplified the model they have used with Amit, and we just use neurons that are inhibitory, so like here, so each neuron. So we took like 5,000 neurons, we connect them randomly by 1,000 synapses, and then we try to see what happened. And so that's what we saw. So that was quite nice. Each neuron was firing like here so these are different neurons. There are probably 50 neurons here, and they are firing from time to time. But if you sum the activity of the 5,000 neurons, you see something that is extremely periodic, okay? So this is, that was interesting. We didn't immediately know what was happening. So there are many time scales in the problem. There is this membrane time constant that I've shown you. There is also the firing rate of this neuron. And after thinking a little bit, we realized that actually what was happening was really quite simple. It's just a feedback inhibition with a delay. So the delay is actually given by what is happening at the synapse. It's not instantaneous. There are synaptic time constant. So when the network is firing a little bit more at some time, after a synaptic time, the neurons are inhibited. It's firing a bit less, and then more, etc. So this is the intuition. So there is inhibition, global inhibition with delay. And it's coming from really the summed action of all these neurons. So that was the intuition, and so we went and tried to explain this a little bit more precisely. So actually we introduced a function that had been introduced much earlier by Bruce Knight, and the idea is just a linear response. So we put some oscillating current in this neuron. They are modulated by this oscillating current. If we have many neurons, some activity is modulated. So we can compute the modulation by just linear response theory, the modulation of the firing rate given by some synaptic current that is modulated in time. And then, as is very usual in statistical physics, we can try to do that self-consistently. So first of all, we can really compute this response function looking at a neuron that is submitted to some mean oscillating current plus fluctuation, this using a Fokker-Planck equation. So we can compute this response function. And so when the noise is low, this is very peaked at the firing rate of neuron. But when there is a lot of noise, like in the brain, it's actually absolutely no trace of the firing rate of this neuron. It's some function. But then, if we try to do this self-consistently, we say, okay, there is some current that oscillates. After the synapse, it will be transformed. It's like a filter. And then again, passing through this response of the neuron. And if we do that self-consistently, one can both determine the oscillation frequency and the criterion for oscillation. And what is happening is that there is a threshold for oscillation. The inhibition should be strong enough so that it oscillates. So that's nice, but okay, there is a famous sentence from Thomas Huxley, who is also a well-known biologist, 19th century of biologist, who, as you know, say that the great tragedy of science is the slaying of a beautiful theory by an ugly fact. So to try and avoid this, or maybe to see it, we teamed. It was great to team with our colleagues and friends at LPENS. So people at LPENS, I mean the neurophysiology group, as you will see, probably in the rest of the talk, is very much in love with the cerebellum. The cerebellum is a structure that is a small brain in the back of your head, and it's a structure that is involved in learning, at least learning movement. Okay, and so Adrian, since he has recorded from many structures in the brain, had recorded from the cerebellum. So here is the cerebral cortex. You see something that is at 60 Hertz, is a very famous gamma oscillation. But in the cerebellar cortex, in the cortex of the cerebellum, you had noticed that there is this 180 Hertz oscillation. So Clément, Léna, and Camille were studying the cerebellum, recording from rats in vivo. So we decided with them to try and understand this oscillation. So here I show you a bit more the cerebellum. So basically the cerebellum is nicer to study than the cerebral cortex because it's very crystalline. So there is this very nice Purkinje cell that is here, is aligned in a very crystalline way. And then there are some input that comes from this granule cell that come here in parallel fiber with this Purkinje cell. And the Purkinje cell is the only output of the cerebellum. So it's a nice structure. And so this is a Purkinje cell that was imaged by Boris Barbour. It's very nice. And since we'll speak a little bit about Purkinje cell, I show you Jan Evangelista Purkinje, who was a Czech neuroanatomist. Okay, so what did Camille and Clément saw? They saw something very similar to what was told by Reinhardt, Ecole, but with much more precision because the technology had evolved quite a bit. So here what you see is all these Purkinje cells, and these are the single units, so you have a lot, the firing rate actually is very dispersed. It goes from 0 to maybe 150 Hertz. But if you look at the single unit spectra, you don't see any oscillation. If you put them together, or if you record with an electrode, which is the sum activity of all these neurons, again you see a peak at 200 Hertz. So that was very nice. Okay, and these two peaks are the same. So here it's clear that the oscillation is apparent from the sum activity of many cells. Okay, you don't see it in single cells because it's too noisy, but if you put enough together, like in the simulation, you see the simulation. So actually, so we could, so this is measured, okay? We can measure the synaptic current, which gives rise to this frequency. We know the rise time of the synaptic current, the decay time, the peak conductance. And okay, so we had a simple theory that was what we saw. Okay, so that was in simulation, that was not too bad. But, okay, the oscillation frequency was okay, but we had a problem because the synaptic conductance, you remember that there is a threshold for oscillation, and in the experiment, the peak conductance was like 0.75 nanosiemens, and it was really like 3 times below threshold in our simulation. So, okay, we were a bit puzzled by that, worried actually. And then Boris said, okay, you're nice, you're theory people, but you know, the Purkinje cell is like this. I mean, it's maybe not such a good approximation to say that it's a single compartment as we were doing. So, okay, so we said, okay, you're right. Maybe we'll try to do the next thing. We'll put 2 compartments. Okay, so we tried that, and this worked beautifully, actually. So then, okay, if we do that, for some reason, using 2 compartments, removed the, went down, the threshold went down by like a factor of 4, and then we could have a model. So this is what is measured in experiment, and that what the model is. So it's very, very similar to the experiment. Okay, so that was fine. But then we had to explain somehow what this threshold was coming from. So actually what we discovered with Boris is that there is a resonance in this cell that is really induced by the cell morphology. So as I said, we just, instead of 1 compartment, we modeled that in just the next complication is just, so we have the soma here, and we put all the dendrites together, and there is a junctional resistance here. So a circuit that is just slightly more complicated. But when we do that, and there actually a resonance here that is with the parameters that are known, which is about 200 hertz. And so the thing is that this junctional conductance gives additional time scale, and then by analyzing this rather simple thing, we could get what is the resonance frequency. And then Boris, so it's very nice to be here, because Boris really tried to measure this resonance in single Purkinje cell, and this is what he obtained. Okay, so that's the phase, and that's the... So I should say that these resonances are rather broad. I mean, they are not like quantum electrodynamics or electrodynamics in a cavity. I mean, okay, so there is a small resonance, but it's enough to get above threshold, and so that's what we published. Okay, and so one important point here is that you see that, okay, the cell itself has adjusted to the dominant frequency of the structure. So the cell has a resonance at 200 hertz, while there is an oscillation at 200 hertz. So it's kind of interesting. And this is the case in many other areas. Here it's a recording from hippocampus, where there are 2 frequencies at 40 hertz, the gamma rhythm, and around 8 hertz, the theta rhythm. And here also, if you look at different cells, you have resonance that are like maybe around the gamma rhythm. Here it's the membrane potential, and here these one are around the theta rhythm. So the view from this is that, okay, there are these rhythms, and the cells are really tuned to this rhythm. Okay, so we tried also to explain that, but I'll spare you this thing. I will just tell you a little bit more what we're interested at the moment. And the idea is to look at spatially extended things. So this is, first of all, it's another regime of oscillation. It's a beta oscillation, and these are things that have been observed for a very long time to occur during preparation of movement. So if, for example, people use this task in monkey, where monkey had to get a reward, had to push a lever, and there is an instruction, and then there is a delay, and then a go signal. So the monkey has to wait not to push too early the lever to get the reward. And during this delay period, there is this oscillation at around 20 hertz. You see, so here is the go signal, and you see this big oscillation. So you see that the frequency now is around 25 hertz, so it's lower than this 200 hertz oscillation. And so, so this is, maybe I come back to this. So this is coming from, as we will see in just a minute, from a circuit where there is, again, feedback inhibition, but going from excitatory to inhibitory neuron in a loop. Okay, but the nice thing about this, which was discovered more recently, is that actually these beta oscillations are not often are not synchronous over the motor cortex, but they travel in waves. So here, this is a recording from more than 10 years ago. But so there is a wavelength and a speed about 20 centimeters per second, and you can see the wave here. I mean, the excitation is going there and there. Okay, so and here is a kind of recording. So this is the kind of thing we'd like to understand. And so for this, we have used a strategy that is very classical. So as I said, the idea is, which goes back to Wilson and Cowan. So they, in the 70s, they tried to describe neuronal population by mean field equation like this, where you have you describe what is happening in a typical excitatory neuron and a typical inhibitory neuron. So these are the current that go into the neuron, and these are the firing rate of this neuron. So you just say that the firing rate is proportional to the current that enters the neuron. And so these are very simple 2x2 equation. With this equation, you can get oscillation and what you like. But for a long time, these equations were nice qualitatively, but it was difficult to make contact with, say, a specific simulation of spiking neuron or specific neuron. But more recently, people have realized that if one chooses appropriately this function, this firing function, and if one makes this time adaptive, that is, if they depend also on the current, then it's much easier to describe. I mean, one can get very close to simulation of more complicated spiking network. So here, for instance, you see for this very simple thing, something where there is the network simulation, and here this simulation of 2x2 equation, and here there is not only this, but a noise term that is proportional to the square root of that. So this resembles a lot what one sees in the spiking network. And if one compute the correlation of activity in the network and in this modified thing with FAT, they go almost on top of each other. So this is a framework that is useful to look at more complicated things. So we have looked at the moment at chain of this thing where we put coupling that are like exponential, and this is measured, so one can look at this thing. And what we have observed actually is that if we, so normally excitation is, inhibition is supposed to be short range in the brain and inhibition more long range. And with, if excitation target only excitation, what one sees is something that is very familiar to people in nonlinear dynamics, is some spontaneous instability of the phase. So all the oscillators do not oscillate in phase. There is a, if one compute the phase equation, there is a negative diffusion coefficient, which is restabilized by the positive one. So there is a spontaneous oscillation modulation that appears. And this depends really on the topology of the connectivity. If the excitation target the inhibition, then there is no instability. Okay, so, and then even if there is no instability, what is happening is that since there is noise in this network, these oscillators, there are spontaneous phase differences that are created by noise. And this is very similar to things that people love in statistical mechanics, like the, say, KPZ equation, where you have noise on the, here this is really the phase of the oscillator that is pushed by noise, and that is wandering. So when one compute this, we can, the idea is that, okay, this phase gradient gives you apparent propagation, and one can compute the apparent propagation that we see. So in this very simple model that I describe at the moment, we have a velocity of propagation that is higher than what is seen in experiment. It's not, the desynchronization is not enough with noise. And we think that is maybe due to delay in this coupling, so we are investigating that. And what one sees is, actually for the moment we have just looked at it in one dimension, but there are more things that are observed, like this plane wave here, radiating wave, rotating wave, and also this wave, I mean the planar wave, have privileged direction on the motor cortex, which is maybe related to anisotropy in the connectivity. So there are still things to understand. So in the rest of the talk, I will try to describe you another aspect that we have studied, which is probably the one that is the most interesting, if you think of this, is learning. And so how learning and synaptic strength are related. C'est pas grave, Julia. Okay, maybe. Okay, thank you. So of course, jusqu'à quelle heure? Okay, so I will try to describe you what we do with learning. So again, we work in the cerebellum, because cerebellum is a main site of motor learning. So I show you a very simple example of motor learning. Is if you, so usually there is something that is called vestibulo-ocular reflex. If I turn your head, your eye will go in the other direction, so that to maintain the gaze on a fixed target. So here it's depicted. There is a monkey. If it turns the head, it will always be on the banana here. Okay, the head, the eye are compensated. But then if you put prism, like on monkeys or even on volunteers, at the beginning, when you turn the head, it's not really compensated, so there is retinal sleep. And after a few hours, then it's compensated again. So the gain of this reflex has been adjusted. Okay? And this is dependent on the cerebellum. If you have cerebellar lesion, you cannot adapt the gain of this thing. And nowadays, people also look at this in the, say, in a mouse, where you can do much more. You can target specific cells with genetic manipulation. Okay. So then, what is the current theory for this learning? It's rather simple, but it was proposed, I mean, it's simple, but once people have told you that. So it was proposed by David Marr, a very talented neuroscientist. So it was proposed in '69 when Marr was 24-year-old, and he died at 35, very tragically, from cancer. So this is the current theory. The idea is that all these Purkinje cells, which are the only output, should adjust their firing rate depending on input. So they receive this input here via the parallel fiber, and so they have to adjust this synapse, which is here as depicted by Boris. I mean, this is a real thing. This is the Purkinje cell and the parallel fiber. So this synapse should be adjusted. And the idea of MAR is that there is another structure which is very striking, which send fiber here that wrap around each Purkinje cell. So each Purkinje cell receives some input from a cell in the inferior olive. And the idea of MAR is like the old teacher. If this Purkinje cell fire when it's not good, then it will receive a big stimulation from this fiber, a little bit like the teacher with a ruler sticking your finger. Okay? So this is the idea of learning. Okay, so if this is, and then, okay, this synapse is depressed. So, okay, that's the theory that was worked out, I mean, proposed by Marr and also by Albus, and there is some evidence by Ito, a Japanese person. Marr is a computational neuroscientist, Albus was a mathematician, and Ito is a well-known neurophysiologist. Okay, so Boris said, okay, since this is the main site of plasticity, let's measure this synapse. And so that's what he obtained. So what he obtained is that, okay, so he did this with his student Philippe Izop. And what he obtained is that, okay, he stimulated, like, he measured 4077 synapses. And what they found is that only 34 of these 477 synapses gave some current, so were active in some sense. All the others were silent. So that was a lot of work, and you see the 37 that are non-zero, a lot of work to find a lot of silent synapses. So just as I said at the beginning, we met completely by chance in a Chinese restaurant that no longer exists on Rue Claude Bernard, was cooking good soup, and we were just sitting at a neighboring table, and Boris explained us this and told us, okay, you are a theoretician, so you please explain that. Okay, so that was a bit tough. So the idea is, okay, to go back to Marr. So Marr proposed that the Purkinje cell is working like a perceptron. So what is a perceptron? A perceptron is something that classifies things into two categories. So you have fiber, you have input here. So like if you have picture in black and white, if you would have 1 if the pixel is black and 0 if the pixel is white. So you feed all these pixels into this machine. This machine takes the sum of the input with weight. And then if the sum is above some threshold, it will fire something saying this picture is a picture of a physicist, and will not fire saying this picture is a picture of biology. So it computes the output based on the weighted sum of the input and comparing to a threshold. Okay, so that's the model of the perceptron that was proposed by David Marr. And so this model, in fact, was studied in the 50s, was proposed first by Frank Rosenblatt, then studied by Marvin Minsky and Papert, and then by many people. And also by many people here in the 80s, because it's very related to the soft-field model. So this classic learning problem can be really addressed well by statistical physics technique. And Elizabeth Gardner in particular made a great advance on this. She showed how one can compute the capacities, how many association between picture and label one can learn. And the idea is really inspired by the physics of disordered system. One can compute each association is a constraint, and one compute the typical volume of that remain to adjust the synapse after all these constraints. And so this has been worked out by Elizabeth, and there are many contributions here and elsewhere that have looked at this problem very extensively and computing the the capacity, how many associations can. So we just took this approach and computed what were the synaptic weight if the Purkinje cell learns as much as it can. So that's what we did. And what we realized is that since the inputs are excitatory, when you learn, actually you increase or decrease the weight, but you cannot, the weight are bounded by 0. They cannot go negative. And so when you learn a lot, there is a lot of constraint, and many synapses have 0 weight. So we could compute that, I mean, using the technique that were developed. And we found actually that there is at least 50% of weight that are 0. And then if there is, so if there is some margin of robustness, we say that, okay, things that should be 1 are bigger than some margin, and some that the sum here should be bigger than the threshold by a margin, or less than the threshold by a margin. If this margin is increased, actually the number of silent synapse increase. So we could compute, we could actually compare with the experimental data and propose an explanation for this data, saying that, okay, the Purkinje cell learns as much as it can. So this is fine. And recently we came back to this question, which is the question of learning and credit assignment. So the whole idea of this Purkinje cell learning is fine, but then one can wonder, there is this teacher, this fiber from the inferior olive that tells the Purkinje cell what it should do in some sense. This is supervised learning. And the question is, how can it, how does this fiber knows that? So this is a famous problem of credit assignment that was stressed by Marvin Minsky. So I can explain this in the example, say, of learning a complex movement like walking or riding a bike. When you try to learn riding a bike, you get a very poor, as in many usual tasks, you get a very poor feedback. You just know that you fall or not. And then you have to adjust a lot of, there are many muscles involved, so you have to adjust a lot of neuron, of muscle to actually ride this bike with just this information that you are falling or not. So this is complicated, and this is this credit assignment. How should I adjust the muscle to avoid falling? So one solution that actually Marvin Minsky proposed originally is to try and do that by trial and error, so that if you have the task, then you perturb a few elements. If the task improves, then you know that, okay, these are these elements that are responsible, and then you can, and you just change this element. So this is a very attractive framework, and it has been considered in the context of song learning in birds. So these zebra finch, they learn their song from their father, so it's known that they just imitate the song of their father. They learn it and try to do the best, reproduce it the best that they can. And there is one structure that has been identified as a source of stochastic perturbation from this learning. Okay? So we wondered, is actually the classic theory says that the cerebellum learns by this inferior olive, but we don't, I mean, it's difficult to imagine how this precise information is coming from. So maybe it's also working at trial and error. So, oops. So what we propose is actually that these cells is not only a source of error, but also a source of perturbation, so that these Purkinje cells actually are perturbed all the time. So these cells, if you record them, they are also always spontaneously active at 1 Hertz, irrespective of what you do. So what we propose is that they are actually a source of perturbation, and that you're learning, because these cells that are perturbed are the ones that you will define. So if you think of this immediately gives some experimental prediction. So the idea is that if you have, so you need a perturbation from this olive to this, and if it arrives at the same time that the input from this parallel fiber, then it will increase the rate of the Purkinje cell. And if there is no error that is signaled, it means that, okay, it was good to increase the firing of this cell. So you would predict that in this case, the synapse will be potentiated. And if you have increased thing here and then there is an error, the synapse will be decreased. Okay, so this is a very simple prediction that can be tested in slices. It's quite different from the classic idea, where the idea is you depress the synapse if you have an error, but you don't care what is happening at this time. So Boris and his student, so there are some detail of how they did the experiment, but in any case, you do this. You do this plasticity protocol where you stimulate both this climbing fiber and this parallel fiber, and if you stimulate it at the same time without error, what you see is that the strength of the synapse, so this is before and this is after, it increases, and if you stimulate it with an error after, then it decreases the synapse. So this somehow is an indication that we are not completely on the wrong track. Then if you want to implement this, you have what you want to do is to have your current performance somewhere and compare it to what you are doing when you are perturbing cells. So you need to monitor the performance. And so since this inferior olive is giving this error, what we propose is that the performance is monitored by inhibition in the inferior olives. If the error is bigger than this inhibition, the inferior olive will fire and will give an error, and otherwise it won't. So this is the way we give this perturbation. And then, okay, so we have some framework, and what we looked at is a very simple model where we have to adjust the firing rate of many cells given a global estimation of this error, and all these cells have to go to their target. So if we do that, we can show that this thing can work. I mean, this algorithm where you perturb cells, you adjust them, and you have only the error that is coming from the global firing of the cell is working. So I can show you that a little bit. So it's a bit technical. Bit late, so I won't go into that, except that it doesn't want to go in the next slide. Okay, so a bit block. So it will come. Okay, so if we have many cells, then okay, they can go to their target with this algorithm and things are fine. Okay, so we have this theory at the moment. There are many things we have to improve. Boris is looking for in vivo data to see if this is working really in mouse that learn the motor task. And maybe there are also other areas where we think that maybe this learning with a perturbation is involved. Okay, so I'll try to, so this is the conclusion. So I've shown you actually two things that were actually quite different. The first one was oscillation. So it was a very dynamic view of the brain. There was a key role for recurrent connection. There is a global dynamics. And the neuron was diverse in the sense that they did not receive the same input, but there is not much specificity. The second part was rather opposite to that. There is not much dynamics. It's just recognition. This is mainly feed-forward. There is not much recurrence there. And individual neurons, they adjust their weight. They have specific role to recognize things. So this seems like a too disconnected view. And so clearly at the moment, what we, in a perspective would be to try and match this thing, and this is what is done by many groups in the world, is trying to record numerous neurons that is possible now with the progress in molecular biology to mark a lot of neurons at the same time, optogenetics, while an animal is learning and performing diverse tasks. And I hope that in the future we'll arrive to match this thing and to understand better how the whole brain works in an integrated manner. So to conclude, I'd like to thank most of the people that contributed to what I told you. So prominently Nicolas Brunel, Boris Barbour, Jean-Pierre, Jonas, Magnus, Serdian, who is here at DEC, Jonas and Serdian were postdoc with me, and now they are permanent. Jonas at LPENS and Serdian at DEC. Then Eli is starting his PhD, and so some of what I've told you was done by Camille de Solage and Clément Léna. We have also collaborated with Stéphane Dieudonné, Guillaume Duguay, but I didn't tell you about that. Guy did this remarkable experiment about the synaptic plasticity, and we have other projects with Herman and Antoine Triller. Thank you.
Coming back to the first part, oscillations. Assume you construct an artificial network, but you add by some mechanical mean an oscillatory behavior. What does the network, what is it that it can do that it could not before? Or is it a nuisance?
So that's actually a very good question. It's actually the main question in the field. There are proponents, I mean, people that think that's a nuisance. Okay, you have a bridge, you walk on it, the bridge oscillates, but has absolutely no function for the bridge. It's maybe detrimental, and then you have epilepsy, and that's really bad. Now there is a lot of experiments that show that at least correlational evidence that these oscillations have some importance. For example, there are a lot of experiments of attention. If you show target with attention, it seems that your attention is modulated at a 10 Hertz rhythm, for instance. So that if you, I want to show you a target in some phase of the oscillation, you don't see it as well that if it's shown on the top of the oscillation. So there are a lot of evidence like this. For example, also, if you record in the visual cortex, and if you attend the stimulus, the amplitude of the oscillation will grow a lot as compared to an unattended stimulus. But at the moment, there is no definitive answer to this question, and it's really something that needs to be clarified.
Yes, I come back to your first part. You simulated this by circuits with only capacitance and resistors. And so you have, but yes, but you do not include any inductance in it. So we know that the brain produces magnetic field. You measure electroencephalography with Josephson junctions. So there is probably capacitance in it if you produce magnetic field. And capacitance with an inductance gives you a resonant frequency.
So actually, that's the part I skipped. so actually, the resonance is coming from an inductance, but not from a magnetic field, it's thought. These conductance depend on voltage. They are called voltage-gated conductance. So when you compute this, I mean, this dependence of the conductance on voltage give you a resonant frequency, which is a bit like an inductance. If you linearize things, it gives rise to an inductance. That's just coming from the derivative of this dependence. So actually, that's how one gets this resonant frequency in neuron. And something we studied, I did not talk about it, is how this resonant frequency impact this oscillation. And how can you see it? So now the real magnetic, the magnetic field or even the electric field, how it impact neuron at a longer distance, is thought to be a weak effect, but it's debated, at least for the electric fields, what people call emphatic interaction. In some cases, maybe it's important, but usually it's supposed to be a weak effect.
Another question? Oui? Micro.
Thank you. Excuse me. In your model, can I ask my question in French, please? Okay, thank you. Depuis quelques années, en neurosciences, on essaie de mettre en évidence le rôle de cellules gliales dans l'apprentissage. Est-ce que vous pensez que ça pourrait apporter des affinements intéressants à votre modèle?
So glial cells are interesting for different purposes. First of all, because they are a very important role in pathology. When you have demyelinated cells. They are also interesting because they have calcium waves in glial cells. Otherwise, for rapid things like this oscillation, I'm not sure they are so important in the sense that they don't seem to have this really fast response in some sense. But they are certainly important at the synapse since they recapture things. And so I don't know. We have not really studied a lot glial cell at the moment. And actually, there are not many people at LPENS that study glial cell. There are people in other institutes.